Well, you guys tell me when you think there are enough participants in.
Yeah, I will start the introduction soon. Okay. Hello, everyone. Welcome to the our last special lecture series. So my name is Min Zhang, and I'm a professor of statistics and also the associate director of data science at Purdue University Center for Cancer Research. So this seminar series is part of our NCI-funded R25 workshop on big data training for cancer research. So today we have three panelists, John Fry, Nadia Lama, and myself. So John has helped with setting up the Zoom meeting and handling all the registrations. And he will also record the lecture and post this recording on our workshop website later. So now I would like to introduce our webinar moderator, Dr. Nadia Lama. So Dr. Lama is a research assistant professor in the Department of Comparative Pathobiology at Purdue. She has been the manager of the Purdue site of the Collaborative Core for Cancer Bioinformatics since 2015. She has a background in biochemistry, molecular biology, and computational biology, and Dr. Lama actually got her PhD in 2015 from Purdue. So her research interests include single-cell data analysis and data integration with a focus on immunological diseases of the prostate. So with that, I will hand it over to Nadia.
Thanks, Min. Welcome back, everyone. So just as we've done previously, you'll remain muted for the duration of the seminar, and you can use the Q&A function at the bottom of your Zoom screen to ask questions. Feel free to upvote questions that you're really interested in hearing answered, and those will bubble to the top. And I believe we will be taking questions sort of sprinkled throughout the seminar and demo. So with that, I'll hand it back to Min.
Thanks, Nadia. It's my great pleasure today to introduce our speaker, Dr. Sean Davis. So Dr. Davis got his BS in mechanical and aerospace engineering from Princeton University, and then his MD and PhD from the University of Pittsburgh in the School of Medicine and Public Health. So he completed his residency in pediatrics in Children's Hospital and Regional Medical Center, followed by pediatric hematology oncology training in Johns Hopkins School of Medicine. Oh, he's actually joined with NCI fellowship. And then I think he stayed at NCI for how many, like 13 years or maybe more than that, before he recently moved to University of Colorado School of Medicine, where he currently is a professor of medicine, and his research interests really at the intersection of public data resources, software and tool development, and cloud technologies, and their applications in biomedical data science. So in this course, we have used a lot of different packages from our bioconductor, and Dr. Davis is one of the major players in bioconductor. And we also talk a lot about different types of big data, over the past 2 weeks. I think today is really, the time that Dr. Davis will speak on how we can use this, public high-throughput data sets to help with our research. So please join me in welcoming Dr. Davis.
All right, thanks, Min. And thanks to the whole team for the invitation. This is, I think, my 4th or 5th year now talking to Purdue. Unlike many of the students, I've actually been to Purdue. Imagine that, right? Actually traveling to give a talk. I'm hoping that we can keep it interactive, even though I happen to be in Colorado, which for those of you who don't know, Colorado is, it is 11 a.m. and my talk was on my calendar at 3 p.m. So you're going to get a very interesting mix of slides, some hands-on, and And I'm hoping that that's going to lead to a lot of interactivity. So let me start, I guess, with some slides. I'm going to share my screen here. So I'm assuming at this point that you all have been working with Bioconductor and have seen some of the Bioconductor capabilities and used it for data analysis. For those of us who generate data, Bioconductor can live by itself on your laptop, and it's quite usable, and that's kind of what it was designed for. But we live now in an age where we live in a sea of biomedical data. And I've drawn Bioconductor here in the middle of the slide, just to highlight it. But if you think of each of these boxes as a node in a graph, and have you guys talked about, I think you did your first network session yesterday, is that right? Okay. So if you think about this as a node, or as nodes in a network, I haven't drawn on here the lines, the relationships or the edges in the graph, but there are a huge number of edges between each of these boxes and each other. And Bioconductor sits in the middle here and has an edge drawn from each of the boxes on here directly back to Bioconductor. And that just means that as you start to think about your own data and research, there are ways built into Bioconductor that allow us to connect directly to these external resources. And what I want to spend some time talking about is sort of the nuts and bolts of how Bioconductor talks to these other systems. And then I'm going to end with a little bit of work that's relatively recent, where we actually explicitly tie some of these resources and data together in our own sort of developed network to help understand public data. So I guess I kind of described it already. Bioconductor is a vertex in the omics data ecosystem. And I'm going to talk a bit first about the cancer data ecosystem. I'd asked people to raise hands, but do we have a sense of how many people consider themselves sort of cancer researchers? Is it above 50%, do you think? You guys think?
I think all of them.
All of them, okay, all right. So you guys have heard this before. I'm just going to go through it quickly, just because you may not have heard it in this context. Bioconductor is large. It's an open source software community. Open source means literally you can download the code and reuse it as you see fit. And it's dedicated to the analysis and comprehension of high-throughput biological data. We used to say, instead of high-throughput biological data, it used to say omics. Actually, it used to say genomics data. And over the last few years, that transition away from thinking about everything as genomics to really all kinds of high-throughput biological data it's ongoing, but certainly well established. I always make the joke, Min's seen this many years now, that I'm going to spend the rest of the time reading through all the packages that are available in Bioconductor. They're in alphabetical order, so no, I'm teasing. But I allow you guys the time and energy to do this if you want. But I'm going to start with this really foundational concept that in order to perform reproducible research, we have to have sort of common ways of thinking about our data resources and also about our software algorithms. But Bioconductor, if you sort of spend a lot of time thinking about it, one of the main successes with Bioconductor, and this establishes that it's quite different than say the Python world or the Julia world, is that we rely heavily on these software data containers. So if we think about what the kinds of data that we get out of these high throughput omics, or these high throughput biological experiments, they're typically very similar to each other, at least conceptually. So we have samples, we have measurements. In this case, I put the name features or genes for the measurements. And then we have the actual numbers associated with those things. So by samples here, I mean, we don't typically just have the name of a sample. We also have information about whether the gender associated with the sample is male or female, or whether the cell type is epithelial or endothelial, etc. And same thing for features or genes. Those could be genomic locations. Those could be proteins in a mass spec experiment. They could be antibodies in an immunoassay. And then the assays themselves are typically quantitative, so they're numbers. Those could be integers in the sense of counts for RNA-seq or single-cell RNA-seq, or they could be continuous variables, as in the blood chemistries of patients. So we need to recognize the complexity in high-throughput biological data. As we think about experiments, they're typically, that is, experiments involving data, they're typically not one-shot deals. We want to have version control. So we want to keep track of all the versions, not just of the data themselves, but also the software that's used. Bioconductor relies heavily on continuous testing and integration, which means that if we change something, we want to know that that change doesn't break things. And it's a good sort of concept to develop in our own workflows and our own data analysis, to have ways of sort of testing that, sanity testing what we're doing. As we think about reproducible research now, moving a little bit back from Bioconductor, having a text-based workflow is really important. So a text file that leads to capturing the analysis that we're doing directly. No GUI, no points and clicks involved. And as soon as you involve a point or a click and you don't capture the result of that, or the choice that was made, you have an irreproducible workflow, in the sense that it's really hard then to hand that to somebody else to use. Literate programming, I imagine you guys have been using vignettes or IPython notebooks, Jupyter notebooks throughout. That's a key component of reproducible research. Education is education, and I should add documentation, are both key pieces of reproducible research. And then Bioconductor and the data resources that we're going to be talking about have lots of mechanisms that lead to this concept of fair data sharing. I'm going to talk about that in a bit. The use case that I want to talk about in the cancer sense is the Genomic Data Commons package. This is going to be sort of, I would say, representative of approaches to accessing data. Anything at NCI right now, in terms of data availability, of publicly available data, most of those projects rely on this data commons framework, the same data commons framework that Genomic Data Commons relies on. So the imaging data commons, the proteomic data commons, those things rely on the same underlying infrastructure, and a lot of the same ideas that you'll see in these slides are going to be reproduced in other data commons. So I'm going to start out with a simple goal to explore the somatic variance seen in cutaneous melanoma in a reproducible, reusable way using bioconductor tools. everything that you're going to see in terms of the slides and the understanding of the slides is encapsulated entirely in these few lines of code. So using the Genomic Data Commons package, we're going to find and download variants from TCGA, cutaneous melanoma data set. And then we're going to take those data directly into this software package in Bioconvector called NavTools for analysis and visualization. And like I said, the code to do all of this is included on this slide. What we're going to end up with is a picture like this. That's where we're going to end. And what I'm going to show you is how the Genomic Data Commons package in Bioconvector converts this concept that we typically use, which is to go to a website, do some pointing and clicking and downloading into code. So what I'm going to be doing here is a set of steps where I'm going to show the code with a box around it. And then the next slide is going to be what's going to be happening if we were going to do this in a web browser kind of framework. And the reason there's a direct one-to-one mapping, that is literally the points and clicks on the website are directly related to code, is because they're both being driven by the same application programming interface or API. So the API is the way the computers are talking to each other. In this case, one computer is my laptop with computer code running in R. And in the other case, it's my web browser talking to a server sitting at University of Chicago. But they're both speaking the same language. They're just using different connectors or different interfaces. So we're going to start with files. We're going to look for files. And the reason we're looking for files is that we want to find this cutaneous melanoma data. So we're going to, the files call maps directly to a files view of a website. And we're going to choose on the left-hand side the cutaneous melanoma project. And that maps directly to this cases.project.project ID equals that, same thing. We're now going to filter the data using the website to pick out somatic mutations, mutex calling, and the data format is a MAV file. You guys talked about VCF files yet? Okay. So in the same way that the gene expression data have multiple samples and measurements on multiple samples in a format, typically like an Excel spreadsheet, there's a separate format for describing variants in somatic variants that are arising in the tumor. And there are actually a couple of them. One of the most common used in the TCGA project is the so-called MAV file, mutation annotation format. It's another tab delimited text file, but it's specifically designed for holding variants. The reason I mention it here is that because it's a relatively standard file, Bioconvector has a way of ingesting those files and then loading them into a data container, not unlike what you've seen with summarized experiments. And then from that data container, we can use a set of standard operations to make plots, etc. So this slide, those 3 clicks, equates to this little block of text. That red block there describes the actual file. And in this case, it's just one file. We're going to grab the ID of that file and then add files to cart and download is this last step. So essentially, those few lines of code represent the 10 or so clicks, and as well as the download. And these data will then end up on your laptop. And we can then move on to using math tools. So math tools is, you all have used DSeq2, I imagine. Yeah. So if you think of math tools as kind of the DSeq2 of somatic variance, with a somatic variant file, a MAF file, we can, like with DSeq2, we can do data exploration, we can do data analysis, we can do plotting, etc. So these somatic profiles from the TCGA melanoma sample, given 8 lines of code, lead us to all the capabilities that are shown here for somatic mutations from the MAF file. I'm just going to show the plotting pieces of that, but you could take this same set of the same few lines of code, and from there you could go ahead and look for APOBEC enrichment estimation, or pan-cancer comparisons, or looking for differentially mutated genes if you had 2 different data sets, etc. So we're going to load the math tools library, and then we're going to read the math file. And then I didn't actually show you the plot function, but it's one line, leads to this. And if you hadn't known that, or if you haven't looked at these kinds of plots before, what's shown here is all the samples across the top here, and there are 467 samples. There's a mutation in at least 90% of the samples associated with one of these genes, and I rank these genes based on their frequency. And again, if you didn't know that this gene BRAF was really important in cutaneous melanoma, you do now, because it's mutated in 50% of the samples. You can imagine now, we did this with cutaneous melanoma, but you could grab all of the different cancer types in TCGA. You could write a loop in R, generate these plots for each of those cancer types, and then quickly hand that to your collaborator or your boss or whatever, and look and see if any of the genes of interest, say for a developmental biology lab, show up in one of these plots associated with a specific type of cancer, which might lead you to a different experimental avenue than you thought of otherwise. There are different plotting types that you can apply here. And each of these leads to sort of different biological understanding. The last one here is the BRAF gene, shown now in transcript space, and looking at where those mutations that I showed before show up on the gene. And you can see that, again, if you didn't know that, most of the mutations are associated with this kinase domain, and there's something obviously very specific going on with this particular location in this kinase. So you can see how with combining the capability of bioconductor to leverage data containers that are specifically built for biological data, and these access routines built into something like Genomic Data Commons, you can take a complex system like the Genomic Data Commons and quickly get down to fairly deep biological understanding, even if you didn't know what you were starting with. So again, coming back, bioconductor here sits in the middle And each of these data resources, I focused on the GDC data portal, the Genomic Data Commons, but each of these other data resources has built-in connectors that allow you to get the data, and then lots of software packages to interrogate those data. So I think my mouth's getting a little bit dry, so let me stop and see if there are any questions at this point.
Yeah, I actually have a question. So I guess, what would you consider, you were showing the plots out of Math Tools about a slide or two ago, what would you consider to be like a high level of alteration? And how many samples would you feel comfortable assuming that an alteration is like not just spurious or being seen by chance?
Yeah, so that's a great question. So I didn't mention much of the detail about what goes into generating a MAP file. The MAP file being the information, the data converted to information about somatic variance. So when we measure somatic variance, what we need to do, a couple of things. We need to subtract out all of the variants that are likely in the germline. So, so germline variants are interesting potentially for cancer predisposition, but they're not typically something that we focus on when we're talking about cancer development or cancer biology. So these somatic variants, we have to then pull out all of the variants that are in the germline. Once we do that, that leads to a couple of things. You can have either a false positive somatic variant, so a somatic variant that appears to be somatic, but is actually germline. Why would you see that? Well, you might see it because in the germline sample, the variant calling in the germline sample, the variant didn't show up. And that could happen because the coverage is too low. That could happen just by random chance. It could happen also because of essentially mismapping of reads back to the genome. You can also have a false negative somatic variant. So a somatic variant that actually exists in the sample, and you don't see it either because it's not well measured. It didn't. There's not enough coverage because in tumors we have an admixture of samples, a mixture of cells. So it could be that the somatic variant is in a very small percentage of the cells, and so we can't see it just by numbers. And then the last is that we may also see spurious reads in the germline sample that result in noise that make it harder to see the somatic variant. So assuming that we do all of that correctly, then we have true somatic variants, true variants that are only in the tumor and not in the normal. Of those, the vast majority of them are going to be irrelevant for biology. So they come along for the ride. In each of our cells, the mutation, there are ongoing mutations continuously, and our immune system combined with apoptosis and senescence protect us from those cells accumulating mutations and becoming cancers. If we're unlucky enough that one of those cells does accumulate mutations and become a cancer, then all of the accumulated mutations that just happen randomly come along for the ride. And so in looking through this set of samples or this set of genes, my guess is a lot of these genes have accumulated mutations that are irrelevant to biology. And probably some of these include significant numbers of false positives. So in practice, each of the variants in here is going to be associated with a set of scores that include predictions of how likely it is that the variant changes the protein, for example. the number of times that the variant has been seen in other cancers, whether or not this gene is known to be an oncogene, etc. So it becomes a little bit of art form to understand what the somatic mutations, for example, associated with this FAT4 gene about 2/3 of the way down. I don't know that gene. I'd have to look it up, and I'd have to go back to the data to decide whether or not I think that that gene is important or not. For some of these genes, ApoB, for example, some of these have associated mutational profiles. That is, they cause mutations across the genome. And when we see this gene active in samples, we might expect that as we look across the biology of the cancer, that we're going to see the effects of this gene being activated or deactivated, as the case may be. I think that's probably a longer answer than you wanted, but the data exploration piece is where things all start. And in practice, where I would use this tool typically is that you have your own data set and you want to pull down data and compare. So you want to add a layer of biological meaning to your own data set. The Genomic Data Commons package would help with that, pulling down these somatic variants for your disease of interest.
Thank you. That was a good answer, so not too long. And then we have another question in the chat. Are there any similar packages to access GTEx data, or does Genomic Data Commons access GTEx data?
Genomic Data Commons doesn't access GTEx data. And that's a great question. Let's, I don't know the answer to that. So let's see if we can find out. I'm assuming you're still seeing my screen. So let's do search for GTEx. Look for accessing 7,000 RNAs. So here's recount. Let's see if there's, here we go. Let me make this bigger. I love these questions, keep them coming. So think about the next one. So Bioconductor has built into it yet another package for looking at genomic data called Recount. The Recount project started 6 years ago, something like that. And the idea was that unlike microarrays, where we had simple text files that described the entire experiment, that is, we got an Excel spreadsheet back that had genes and samples. With RNA-seq, if we wanted to get to that point where we had genes and samples, we had to do this mapping process, and then we had to summarize to transcripts and genes, and et cetera. And that was computationally quite intensive and not something that most groups were able to do at the time. So this group with Leo Collado, Andrew Jaffe, and actually a whole slew of other folks put together a cloud-based project to mine RNA-seq data from public resources. And the reason this shows up in our search is that one of the public data resources that they included in Recount is GTEx. So you can use the Recount 2 package to grab data from the Recount project, and specifically for grabbing data from GTEx. So let's scroll down through the vignette here and see what that looks like. Here's an overview of what they do with Recount. They map everything back to the genome, and then they do some nice counting. And let's see. Oh, and here we're talking about what Recount 2 looks like. They have custom metadata, which allows you to do searches. Then you can download the study and then load. And what do you get after you've done that? You get this thing, which looks remarkably like a summarized experiment, because it is a summarized experiment. In this case, they're showing how when they summarize the data, they summarize it actually not just to genes, but actually to genomic ranges. So you can do experiments with recount2, which you can't do with typical RNA-seq summarizations. And in particular, this little spot right here is, they're trying to show here, is a region of the genome that's not actually annotated as part of a gene, but it shows up reproducibly in their experiments, in the RNA-seq experiments. So these also get captured and can be found as ranges in this summarized experiment. So if you were potentially interested in GTEx data and in a particular gene, you can download, you can find the GTEx data, you can download the study and load it, and now look at the region associated with the gene to see if there are uncommon or unknown transcribed regions associated with that gene. And you can also deconvolve things like rare splice variants, et cetera, from recount2. So for someone who just asked me that question about GTEx, this is a long-winded answer, and it's not the only way that you can get GTEx data from Bioconductor, but it's one that allows you to dig in and really focus on the genomics piece of things.
So then they followed up and said, but recount doesn't provide genotype data, correct? So is there a way to get genotype data?
So the answer is yes, but genotype data is typically, let's see, GTEx.
I love that you're Googling. I think that's my answer to about 50% of the questions.
Oh, yeah.
Usually with Google. So
Yeah. It's I don't even think about it. I don't even think about it anymore. I used to apologize for having to Google, but now, that's the way the world works. So with let's see, with GTEx, protected data and raw data. So genotype data is going to be protected data. And I will put on my bioconductor hat and apologize for the fact that we have not spent enough time in bioconductor thinking about how to access and protect protected data. So what this means in practice is that you'll have to go through the GTEx protected data access process. And then once you do that, then the data that are available are things like BAM files, genotype calls, SNP arrays, etc. All of these things are things that you can access very easily using Bioconductor. So you can read in a VCF file, for example. And in fact, you don't even have to read in a whole VCF file. You can index a VCF file and read in genomic regions or specific samples. And the same thing with SNP arrays. You can read in pieces of it and even do things like imputation. Bioconductor is not necessarily the best way of doing that, but once you have the data though, and read them into a VCF file, all of the tools that you would use for a VCF file for non-protected data, like for mice, for example, work just fine. And some of the map file capabilities that I showed before have non-somatic or germline approaches associated with them as well. So the steps would be go through this protected data access process, look at the files that are available, and in particular, you're probably going to be interested in the VCF files, and then look for one of many packages in Bioconductor for manipulating or using VCF files. One caveat here, these allele-specific expression tables maybe for GTEx, those are often things that people are interested in. So you may actually be able to sidestep the VCF piece and grab the allele-specific expression tables directly. What other questions come up?
Well, if no one else has, how long does it, like, do you have to request access just once, or do you have to do it for each project you're involved in?
Yeah, so you have to access, you have to provide to GTEC and in general for protected data access. There's no blanket statement that any data access committee is going to agree to that says, I want to download the data and use it for fun. And there's a good reason for that. So it may feel like, it may feel onerous to have to go through this process of describing your project and exactly what you want to do with the data. And in some cases, the data access committee, the committee that's going to approve or deny or work with you on your request, may ask questions of you, or may ask you to change things, or may even deny your request. And when I first started looking at these kinds of things and thinking about this, oh man, just give me the data. Just don't worry about it. I'll do fine. I won't do anything wrong with it. The challenge with that kind of thinking is that I've now worked with some patients who have submitted samples to these projects, and they have interests in making sure that one, that their data are used. So if they've submitted data, or their families, in the case of GTEx, have agreed to have data used in GTEx, they want the data to be used, absolutely. And they want it to be used as openly as possible. But at the same time, they want it to be used constructively. So not to do bad things, or not to do things that don't improve research. So that process there is really to make sure that your request for data and your envisioned usage of the data aligns with the patients and the consortium or the community that submitted the data. And that's not so big a deal with GTEx, where the patients are dead. There's a very altruistic piece to that with, for example, pediatric brain tumors, or HIV specimens. Those come attached to kids who obviously can't agree on what to do with their data. So there's a committee there to make sure that the data are not misused. And then for things like HIV, there's a stigma attached. So there can be a stigma attached associated with that disease and with research going on with it. So again, there are good reasons for putting this in place. With NCI data, I can tell you it can take a few days to get access. GTEx, I'm not sure, but my guess is it's pretty quick as well. There are other smaller institutions or institutes at NIH that have different data access request methods that can take longer. And for some, it could take several weeks, just because the data access request process is convoluted, might include an IRB, for example. Those can take longer. Again, long-winded answer, but. Other questions? All right. So I wanted to go ahead and move on then to let's see. Move on a little bit to this public data resources and bioconductor. So for those of you who want to work through some exercises, show you. Well, here's the. I'm going to drop the link in. Can I drop the link in chat? How do we do that? Let's see. Got to find chat now. Sorry, folks. Here we go. As I'm sharing screen, I guess I can't see chat. But it's if you do a search for public data resources with my name, you'll likely end up here. And what this is a workshop that we do every year with in the bioconductor during the bioconductor conference. Actually, we did it 3 times this year because we had 3 different bioconductor conferences. But this workshop is really around this concept of accessing public data. The workshop itself includes GeoQuery, which you may have used already. Have you guys used GeoQuery at all? Not yet. So I'll go ahead and I'll show an example of that in a bit. The Genomic Data Commons package. Then there are 3 other really interesting, very powerful data resources, access resources from Bioconductor for cancer data, curated TCGA data, which is exactly what it sounds like. It's essentially the same data that you can get with the Genomic Data Commons package, but it's been curated, hand curated, to remove duplicates, things like that, and also to make data access very straightforward. TCGA utils for doing mapping between IDs that was common to need to do in early days of TCGA. And then this last package, CBio Portal data. CBio Portal is another data resource for cancer data. It includes TCGA. It also includes the pan-cancer data set and about 150 other cancer data sets. Anything that's in CBio Portal, and I can go to it in a second, you can access through CBio Portal data in a very similar way that you can with Genomic Data Commons. Recount, we talked about. Curated metagenomic data, and phyloseq, and phyloseq is for operating on those data, and HMP 16S data. For those of you who are interested in metagenomic data, curated metagenomic data and HMP 16S data are, again, curated collections of metagenomic data resources. And the thing that's quite different about these data resources and what you can get from public, sort of downloading it from NCBI, is that we've already done the analysis associated with mapping the reads to organisms. So what you get is essentially a table of sample to organism mapping. So it ends up looking a lot like RNA-seq data or gene expression data, tabular. And then the metadata, the annotations associated with those samples are also included. So, for example, you might have a study that looks at premature delivery and the metagenome, so the microbiome of the cervix of folks who had delivered prematurely and those who don't. And so those samples would be labeled with premature delivery or not. And you can do, for example, differential metagenomics, essentially, to ask which species of bacteria might be associated with premature birth. And then the last one here is PharmacoGX. And PharmacoGX is quite interesting. It's looking specifically at the relationship between pharmacology and gene expression. So not only does this have data resources associated with it, it also has a lot of analytical capabilities to model for example, pharmacokinetics, pharmacodynamics in the context of gene expression. I'm not going to go through these in detail, but I don't know if you can see, this goes on and on and on and on. We do this in about an hour without having students do anything. But my guess is if you wanted to walk through this yourself, it's probably 4 or 5 hours of work to just actually step through. But in the process, you'll learn a ton. So that's this public data resources vignette. Let's see what else I wanted to show you. I'm going to jump over to curated metagenomic data specifically. So this is a project that's ongoing. This one's kind of interesting in that we've shifted over to begin to use cloud resources to do a lot of the analysis. So what we do in practice is download data from public resources, SRA. We just talked about SRA at all? Yeah, so we download data from SRA, and then we run a pipeline on it. And what we get back is for each sample, we get back about 25 individual files. We summarize those files, and then we make them available through this R package called Curated Metagenomic Data. And this is just an example of how all the quote-unquote curated X data work. To access metagenomic data, there's one function called Curated Metagenomic Data that allows you to both query and return resources. So in this case, we use Curated Metagenomic Data. This is after loading the Curated Metagenomic Data package. We put in here some essentially free text. And what Curated Metagenomic Data is going to do then is to go through and show you all of the files, all of the data resources that match with that query. And in this case, there's this As Nakar F 2017. This is the name of a study. We can find the, you can actually look it up in PubMed pretty easily just based on that name. And once you find the thing that you want, in this case, we're talking about relative abundance. We want to look at relative abundance, for example. If we set dry run equals to false, what's that going to do? It's going to actually download the data and load it into a new data container that is a summarized experiment, but it's a tree summarized experiment. And why is a tree? What are we talking about when we're talking about a tree here? Well, when we're talking about a tree, we're talking about in the phylogenetic tree, we can summarize the abundance at multiple levels. And what tree summarized experiment allows us to do is to it knows about the relationships between strains, species, genus, etc. We can summarize this tree summarized experiment at any level that we like. So let's say that we want to do it at the genus level. Well, we can do that if we want, and the tree summarized experiment knows how to do that. It knows which species belong to which genus, for example. As soon as you do all of that summarization to the level that you like, then what you end up with is indeed just a summarized experiment. And if the data inside that summarized experiment are if those data are integers representing counts, what kind of data set does that kind of look like that you've been working with? My guess is that you guys talked about the RNA-seq count data, and so they're treated differently than if these were numeric data that sort of followed a bell-shaped curve, more like gene expression or quantitative values from, for example, blood tests. So depending on the type of data, you could then use, for example, DeepSeek to analyze, to ask for differential abundance between two different sample types. Again, we talked about premature birth versus not, but in this case, I believe it's a cancer data set. So asking about the difference between different types of cancer. So again, let me stop there. But the take-home message from this is not necessarily about curated metagenomic data, although if there are folks who are thinking about cancer and metagenomics, this is a place to go. But really to think about this curated data framework that Levi Waldron's group has put together that allows us to access TCGA data, 16S data, or metagenomic data. Questions? Okay. So let me jump then to the last thing that I wanted to talk about, which is kind of where we're going to go next, where we're headed next. So, so I apologize for not having slides, but the idea here is relatively straightforward. Scroll down to figures. So the idea is, so don't focus on the figures yet. The idea is that in the public public realm, there are several million RNA-seq data sets. And if you add on top of those microarray data sets, you have sort of twice the number. So millions of data sets that measure gene expression. And the question that we started with was, there are two questions. One was, could we find a biological signal that was common across these data sets? So data set A and data set B. One has to do with the tumor and a normal, and the other has to do maybe with autoimmunity. One might think that there's an immune signature in the cancer data set that we wouldn't be able to necessarily see because we're not looking for immunity. We're not looking for an immune response in the tumor. We're just, that data set was designed to look at tumor and normal to ask for differential expression between the two. In the autoimmunity data set, we might explicitly be looking for an immune signature associated with patients who have an autoimmune disease and those who don't. Could we take, for example, principal components analysis on the autoimmunity data set? One might expect, I'm making this up, but one might expect that you would see in the first, second, third principal component an immune, an overrepresentation of immune genes in that principal component. If we took that same principal component and mapped it to the cancer data set, would we see it? It won't be in the top 3 principal components, but it might be number 20 in the principal components. Could we see that? That's kind of that's the idea of looking for signals that exist across data sets. And operationally, we calculate 20 principal components on as many data sets as we can, and then we look for correlations between them. And those that have high enough if the principal components are correlated at a high enough level, then we lump them together and we call them RAVs, replicable axis of variation. So RAVs are just principal components that are correlated with each other at a high enough level across data sets that we think that will represent that we're seeing shared biological signal. The second piece is, can we use what I just described to understand a new data set? So in practice, what that means is that we're going to take our new data set, we're going to calculate principal components on it, and then we're going to ask, are those principal components in the new data set correlated with principal components that we've already seen? And if so, how correlated? If they're correlated enough, then we're going to say that the biological signal in the new data set has been seen before. Does that all make sense so far? Okay. Any questions? Okay.
We have one, but it's about metagenomics. So
Okay.
should I wait or?
No, maybe let's wait. Let's hold.
Okay.
Can we hold that one to the end?
Yeah, absolutely.
Okay. So I'm going to talk for another 10 minutes maybe, and then we'll stop for questions and discussion. So in practice, what we do is this. We start with a public database. And then in this case, we started with a database from Casey Green's group called Refined Bio. Actually, it's not from Casey Green's group. It's from the Cancer Data Science Lab, Alex's Lemonade Stand. So that Alex's Lemonade Stand is a pediatric oncology foundation that supports research, and they're very interested in public data. Public database, we find all the data sets that have at least 50 samples in them, because we want to do principal components. We need a large enough number that we can do that. Turns out there are about 1,400 of those data sets that are available for analysis. We do principal components analysis. We cluster those principal components based on what I just described. It gives us, and that leads us to a set of replicable axes of variation. You can think of these as kind of like averaged principal components that have been seen in multiple data sets. Then we can take those replicable axes of variation, these principal components, and we can look for example, gene set enrichment analysis. So what does a principal component look like in practice? Well, it's for each gene, you get a weight, right? For genes that are responsible for significant amounts of variation in that principal component direction, those genes get a higher weight. And for other genes that are not so important, they get a weight near zero. So you can think of this as it's kind of the same problem that we see when we talk about gene set enrichment analysis, just the ranking is done differently. So each replicable axis of variation then gets attached to gene sets that are significantly enriched. So now we've gone from these replicable axes of variation, which are just principal components, and we're now beginning to tie to them gene sets. These gene sets have gene function associated with them, etc. So we're tying biological knowledge to each replicable axis of variation. And then we also, in addition to tying them to at the level of genes, we tie the indices back through the studies that they came from to mesh indexes, or to mesh terms. Mesh terms are the same terms that are used to summarize, for example, PubMed abstracts. So what we did was to take all the descriptions for those 1,400 studies, assign mesh terms to them, and then through the RAV indexes, we assign the mesh terms for principal components for studies with principal components that contributed to each RAV index. So now we have a gene level assignment of biological features to these RAV replicable axes of variation. And we also have a study level through mesh terms. All of that gets put together into this thing called RAV model. The RAV model now, keep in mind what this is incorporating. This is incorporating the gene expression from 1,400 different studies, each of which has at least 50 samples, compressed through principal components, tied to gene descent enrichment analysis. We do all that first, or we do all that prior to making the RAV model. And we also tie it to the mesh terms. This whole thing then represents terabytes of data and millions of compute hours. This can be downloaded to your laptop through this genomic supersignature package that's now in Bioconductor. With new data coming in over here on the left, we can then use principal components analysis. This validation score is looking for the association of your principal components from your data set with principal components with the RAV index. And then this is really cool. We can, for each sample in your new data set, we can assign a score associated with each RAV index. So this bottom line is super cool in that what we do is to convert a sample by gene matrix into a sample by RAV matrix. So now if you have, for example, in your new data, you have two different types of tumor, you can now ask questions based on these sample scores. You can ask for the differences in RAV index between the two different cancer types. And if the top differentially expressed RAV score is now going to be tied back to these mesh terms and gene sets. So even if you had only 3 samples of your 3 samples in each of your different tumor types, what we've done is to allow you to transfer all the learning from those 1,400 data sets to your 6 samples, and to allow you to do differential expression or differential regulation based on all the knowledge that is incorporated in the genomic supersignature RAV model. So what this looks like in practice, or what this looks like at a higher level, is we've built essentially a knowledge graph where we take gene expression profiles in, we produce the RAV model. We can use transfer learning, this idea of applying the RAV model to a new data set. We can look for enrichment, text mining essentially. We can look at a level of gene sets, and these gene sets now are not just gene sets based on your differential expression, but gene sets based on the signal, the replicable signal seen across 1,400 different studies. And we can also match to prior data. So we show you an example of, I'm going to scroll down here to show you an example, a really cool example. So of the kind of thing that we can do. So what we're looking at here is we're asking a really specific biological question. Can we find a replicable axis of variation? So these are just quantitative numbers. These are just numbers assigned back to samples that will allow us to predict the neutrophil count from that sample. Okay. So in this case, this left-hand panel, we have the actual neutrophil count from a study. And we can see that we can find a RAV model. It turns out that it's RAV 1551, specific group of principal components averaged together that is correlated with the actual neutrophil count from the samples. We can then take the same gene expression, the gene expression data from that same study. We can predict the neutrophil count based on the gene expression data. This is using somebody else's model. There's a specific way, a specific model. I don't care what that model is, but it allows us to do that prediction. And you can see that our RAV model follows very nicely with the predicted neutrophil count estimate from other means. So we didn't know that RAV 1551 was a neutrophil signature, but we had a data set that allows us to look to see which RAVs associate or are correlated with neutrophil signature. Now we know that at least in one data set, this RAV 1551 is not only a neutrophil signature, it actually predicts as well as a neutrophil signature predictor that was specifically designed to do that. So we didn't know anything about neutrophil signatures. They did, and we can do the same thing. We can then take that same neutrophil signature and we can apply it to a data set for which the neutrophil count was not even measured. So what we're doing now is taking this Mary's data set. We're looking at the neutrophil count estimate based on this specifically built model and what we see with our RAV model. And you can see that even without knowing what the neutrophil count is, we can make an estimate. So this is what I think of as one of the really powerful approaches here that comes out of this, is that we can use these RAV models in known data sets to define which RAVs allow us to, well, we can attach biological signal to these biological meaning to these RAV models based on actual experiments and quantitative values. We can take those RAV models and potentially apply them to data sets that were never designed to answer those kinds of questions. So just one example, but there's a lot of room for imagination as to how you can use this kind of stuff. And again, the people have done this kind of work before, but what's super cool about what Seun Oh, the first author, has done here is she's made this possible with your laptop. So you can do all of this work just with a laptop. Any questions? I know we have a question about metagenomics, but just to point out that genomic supersignature is available, and there's a quick start in Bioconductor. So if we can I see a Q&A. I guess I figured out how to. I'm going to stop sharing so we can see each other. Access metagenomic data based on the location of the body from which the sample was taken. The answer is yes. The curated part of curated metagenomic data curates lots of different. Let me actually show you. I'm typing. see. I'm sharing my screen again, sorry. So even the curation process is kind of interesting. So what we're looking at here is the actual curated data. Levi Waldron's group, Levi in particular, has pulled together a small army of master's students. He's even got a lead trainer master's student who onboards these students every semester and every summer as well. And what they do is to go through and they start with a study, and then they go through and pull out things like body site, whether there's current antibiotic use, control versus disease, etc., age. So all of these things are pulled out, and all of these become available in curated metagenomic data. So when you access it in the way that I showed you before, what you get is these become the annotations that are in the call data of the summarized experiment. But it is a manual curation process. Other questions?
Yeah, we have one about the genomic supersignature. So how is the computational efficiency about the model?
Yeah, the application of the model is instantaneous. It's as fast as calculating a set of principal components, really. So, I mean, the compute time to generate the model, like to actually do the calculation of the principal components, and then doing the clustering to generate the RAS themselves, that piece takes a couple of days. We do that once, and as new data sets get added, we'll do it again. And then the metadata piece, attaching the gene sets, and looking for gene set enrichment, and also the mesh terms. The mesh terms takes a few days, and then the gene set enrichment analysis is actually pretty quick, a few hours. So.
And then I have a question too, that this might be a stupid question, but I'm just going to ask it anyway. How do you think this would work? I mean, if we work with a lot of canine data that we use as a model for human cancer, and how do you think it would work to compare something like that, or to I guess input that data into your genomic supersignature package?
So Cian has done some work looking at mouse, and I would say it works as we'd expect, but those were sort of contrived experiments. So you can imagine that there's a couple of levels here that you would need to sort out. The biggest one probably is that you have to map the dog genes to human genes. So each of these, I skipped over that detail, but each of these studies gets mapped to a set of common genes. So some studies are not going to have all the genes, and some are going to have more, but we map to a common set. So to the extent that you can do that with dog, and I've worked with dog before, it's not straightforward to map to human, but to the extent that you can do that with dog, you could try it. And essentially what you would then need to do is you would convert your gene expression matrix into a set of samples by genes do principal components analysis, and then you're back in the same place that that you would be if you were looking at human. I would just say you take it with a grain of salt. All of this is human right now. So.
Yeah. And well, and then what about like single cell RNA-seq experiments? And would you need to, I guess it probably work maybe without imputation, but.
Yeah, so again, Seun's tried it with single cell, and for these contrived experiments, it works as expected. But there's with single cell, you're going to have mutation problems that you potentially want to deal with. And the other thing is with single cell data, even calculating a principal component, doing a principal components analysis on single cell data, if you have hundreds of thousands or millions of cells, you can't do that sort of with one line in R. You have to be a little more tricky than that. What might work better, at least what might work more practically easily, is to sort of summarize to the level of cell type. So if you can, rather than think of them as individual cells, if you do have some way of collapsing, that might be helpful. Other questions? Well, I'll stick around, but I do want to thank everybody for their attention. Apologies for not having a beautiful set of slides, but sometimes it's fun to give these kinds of talks anyway. So I hope you guys got something out of it. I think today is your last official day and tomorrow's projects. Is that right?
Yeah, tomorrow is the presentation day, and you're welcome to join us.
All right. Well, then the students better use their 4 minutes now to get working on those projects. Min's a really hard grader, so.
Yeah, thank you so much for the great talk. I think it works out, even better. I remember previous you always give a hands-on session. Hopefully, you'll be able to come to Purdue campus again next summer.
I'm hoping to. Feel free, if Min hasn't shared my email, feel free to reach out or whatever. And if people have follow-up discussions they want to have with me, I'm just a Zoom call away. So.
Okay, thanks again, Sean. And thank you all for coming today.
Take care, everyone.
Bye.
Bye.